Well, good evening, everyone. My name is Christine McCartney. I would like to welcome you to the webinar this evening and introduce myself. I'm the Director of the Australian National Centre for Immunisation Research and Surveillance and Professor at the University of Sydney. And I'd also like to introduce my co-host, Professor Katie Flanagan from the University of Tasmania, who's on as part of the ACID subgroup on vaccines, special interest group, who are also co-sponsoring this webinar along with NCIRS and Sydney University Marie Bashir Institute. I'd like to thank you very much for joining us this evening and swapping us out for the usual ABC News time. And I'd like to begin by acknowledging that we are hosting this webinar, at least where I'm sitting, on the lands of the Darug people of the Great Eora Nation. We acknowledge Aboriginal Torres Strait Islander peoples and communities, and I pay my respects to elders past and present, and particularly acknowledge Aboriginal Torres Strait Islander people as the first peoples of Australia and as the traditional custodians of the land and water on which we live, work and play, particularly valuing the principles of self-determination and empowerment, working towards equal participation and equitable inclusion and voice. So this is part 2 of our COVID-19 vaccination program in Australia webinar series. And we are absolutely delighted to have all of our guests this evening. Just a few housekeeping rules. A video of the webinar will be emailed to all attendees and available on our website in coming days. That's a link to the video being emailed. We will have a Q&A panel, and I'm delighted to say that Andy, although it's very early in the morning, our special guest will be staying for the Q&A panel. So I think that's going to be a real highlight of this evening. And if you see a question or wish to pose a question to the panel, you can enter it into the question and answer tab. And if you particularly like a question that has been submitted, you can actually upvote that question by clicking on the thumbs up beside it. And that will enable our questionnaires or our questioners to ask that question in preference to the panelists. So as I've already alluded to, I'm delighted to introduce our keynote speaker for today and speak a little bit about him first before introducing our first speaker. So our keynote speaker is Professor Andrew Pollard. He's a Professor of Paediatric Infection and Immunity in the Oxford Vaccine Group in the University of Oxford in the UK. So Professor Pollard is the Chief Investigator leading the development of the ChAdOx novel coronavirus vaccine against SARS-CoV-2. He chairs or has chaired the European Medicines Agency as well, their scientific advisory group on vaccines from 2012 to 2020. He currently chairs the UK JCVI, or Joint Committee on Vaccination and Immunisation, and is also a member of the WHO Strategic Advisory Group of Experts on Immunisation. And dare I say it, a long-standing colleague and friend of NCIRS. So our other speakers tonight are Dr. Hao Cheng, Medical Officer from the TGA, and Associate Professor Michael Nissen, Director of the Vaccine Surveillance Section and Pharmacovigilance Branch, also from the TGA. And most importantly, I think up first, I'm delighted to introduce our own Chief Medical Officer, Professor Paul Kelly. And I know you all know Paul as the recently now firmly appointed Chief Medical Officer of the Australian Government Department of Health. So I'm delighted to also know that we've worked with Paul for many years. He's a public health physician and epidemiologist with more than 30 years research experience in infectious disease epidemiology and health system research. He's one of the co-leads of the FluCan system, which I'm sure many of you on this webinar participate in, and that tracks influenza on a regular basis, and has a vast experience in infectious disease epidemiology, particularly respiratory diseases such as influenza. So I think it's absolutely wonderful that we have Paul's leadership at this most critical time with the COVID-19 pandemic. So with that, I'll take great pleasure in introducing Paul and inviting him to say a few words before we go to Andy. Thank you.
Thanks, Christine, and it's a pleasure to be here. I'm on Ngunnawal and Ngambri country, and so I'd like to acknowledge the elders past, present and emerging here in Canberra, and to acknowledge the elders and wherever you're sitting, and particularly welcome any Aboriginal and Torres Strait Islander people who may be on the webinar. So we're up to 709 participants according to my clock here. Look, I won't go into a great deal of detail because I like, I'm sure everyone else is looking forward to Andy's presentation, but Christine asked me to just have a quick look at the Australian situation, particularly related to vaccines. But I thought I'd start with the contrast that I usually do at my now quite frequent press conferences, and look at the epidemiological situation here in Australia compared with the rest of the world. And I think that's an important thing to do for us to remind ourselves. And it is one of the challenges of our vaccination program as it rolls out in terms of measuring vaccine efficacy when you essentially have eliminated the public health problem, as we have done for many months now. So the world clicked over 110 million cases in this extraordinary pandemic over the last just over a year today, and now over 2.4 million deaths. Just in the last 24 hours, almost 400,000 cases, and clearly that's an underestimate of the actual cases in many parts of the world that don't have the sort of surveillance systems that we have here or the UK or other developed nations. And 11,000 deaths were recorded. Here in Australia, we are now approaching 29,000 cases since we had our first case late January of 2020. And today, only 3 locally acquired cases, and in fact, all of 3 of those are related to the quarantine-related incursion from the Holiday Inn in Victoria. So 5 cases overall, 2 in hotel quarantine from recently arrived overseas cases. The other 3 were on the 3rd floor of that hotel, and that was their day 11 swab. So we haven't had any cases, for example, in New South Wales. I think this is the 33rd day without a single case. We have only 10 people in hospital, only 1 in ICU, and there's 0 deaths so far this year. So an extraordinary difference compared with the rest of the world. To turn to our vaccine program, of course, that will be a vital part of our public health response to COVID-19 here in Australia, as it is everywhere. We've started with 5 main pillars, if you like, in relation to our vaccine strategy. The first was many months ago now to look, as many other countries did in the world, at advanced purchase agreements to get vaccine, to make sure we were in the queue to get vaccines. We looked at a diverse strategy, as many countries have done, and we looked to get 1 mRNA vaccine. We chose the Pfizer and biotech vaccine. We have 20 million doses locked in terms of that over 2021. We looked at a viral vector vaccine, and we'll hear, of course, a lot from Andy about the Oxford AstraZeneca product. That was the one we chose. Of course, these were all at times where we had phase 1 data at best, but we thought to diversify that looked like, and has turned out to be, an excellent vaccine. Crucially for that, the viral vector vaccine, the Oxford AstraZeneca, we have developed the ability to make local product. And so down in CSL, plant in Melbourne, they have made 53 million doses, and as of this week are starting their fill and finish, so getting them into the multi-dose vials, and they will be rolled out in Australia for Australian use from next month. We're also getting a small delivery, a relatively small delivery, but it should be 3 million doses from overseas suppliers, and that will arrive earlier. The actual date of that is still to be determined, but it could be as early as next week, and that will be very welcome. So we also had 2 other advanced purchase agreements for protein-based vaccines, much more less innovative, but very solid platforms in terms of vaccines. The protein subunit vaccines we're very used to using in Australia and elsewhere. So those 2 were the Novavax vaccine that's going through its final stages of clinical trials, and we believe that will be it already is on the provisional pathway for TGA registration, but we'll be getting more the final data for that in the coming months. We have 51 million doses locked into that agreement, and the other one was the locally developed University of Queensland protein vaccine using their molecular clamp technology, and unfortunately we had to discontinue that because of an issue with one of the proteins causing false positive HIV tests, so that's not going ahead. But 3 out of 4 ain't bad, and I think when that was certainly a risk to take, which we took last year to make those advanced purchase agreements with a paucity of data, but we did, and 2 of them have now gone on to the second stage, which is regulatory approval. Unlike many countries, including the UK, and Andy will, I'm sure will talk to this. We didn't go for an emergency approval because we didn't need to. We didn't have an emergency. We've had the virus under control for many months, and so we went through the full regulatory path, and I'll leave that to Michael and to how to talk to, no doubt, in relation to AstraZeneca at least. So we have regulatory approval now, Pfizer some weeks ago, and then AstraZeneca this week. And it's been a big week this week because we've gone to the next stage, the third stage in our strategy, which is the supply, and so the first doses of vaccine ready for use from next Monday are now in Australia. 142,000 doses of the Pfizer and biotech mRNA vaccine. So that will this weekend be distributed around the country for use and starting on Monday. The AstraZeneca, as I said, is coming, but it's not quite here yet, but that will be part of our supply in the coming weeks. So the next phase, of course, the fourth component is the implementation. That's been many, many months in the planning, enormous amount of effort by many people, including those on this call, guided by our ATAGI group, firstly, and thank you for those of you that are here with that, part of that ATAGI group, and also another group that was set up with industry representatives, the scientific advisory group, scientific and technical advisory group on immunisation. So there's some cross membership there. I'm the deputy chair of that group. Brenda Murphy, the secretary of the health department, is the chair, and there are members of ATAGI on that group, but crucially also people that are very used to the whole pharmaceutical industry and industry more broadly. The chief scientist is a member of that group as well, Australia's chief scientist, and that's been really valuable. But I've had a lot of insights. I've learned a lot from that group in terms of the real world of how one procures vaccines, and it's a contact sport. I'll leave it at that. So that's the implementation. So the slide that's up there now demonstrates the very clear guidance we had right from the beginning from ATAGI. And I remember Alan Cheng at a SciTag meeting actually sort of challenging the government to say, what is the strategy? What are we aiming for? And very clear from the beginning, it's been about saving lives. And that's how our strategy of our prioritisation has commenced. It's the people that will be getting vaccine from Monday will be those that are most likely to be exposed to the virus, and those that are most likely to develop severe disease. And so you'll see from the slide there, phase 1A, up to 1.4 million doses with the 2-dose Pfizer vaccine will be starting with those groups, and pivoting towards the border workers, anyone from airline workers through Australian Border Force, the Defence Forces and so forth that are associated with the border, with our quarantine hotel staff, transport workers that are related to that, transporting of people coming back from overseas, are absolutely in the front line, followed by frontline healthcare workers, starting with those at most risk of being exposed to COVID, and then aged care, disability care staff, and residents of aged care and disability care residences. So the states and territories have set up Pfizer hubs. They'll be starting. Those will be delivered this weekend, and the Commonwealth government has specific teams that will be providing in-reach to aged care and disability care from Monday. So that's actually happening, and it's an extraordinary time for us to be part of that process. The final step in that 5-step plan is post-market surveillance, and I'm sure that Christine and others will talk to that later, but that's a crucial component of monitoring what happens end to end with the vaccines that are distributed, particularly looking for adverse events and recording those and investigating those. And I'm sure Michael will talk to that shortly. I'll just finish off with one final statement. That's on the challenges that we face into the coming weeks and months as we roll out this stepwise strategy to vaccinate as many people in Australia who want to be vaccinated by October. And I'd say I think of 3 things in relation to that. The first one, this is an enormous logistic exercise. People have said it's the biggest logistic challenge we've had since the Second World War. I'm not sure that that's true, but because we are very familiar with getting vaccines out to people. But there are logistic challenges in relation to the Pfizer vaccine. It's -70 requirement, amongst other things. But that's there. But just the scale of trying to get people vaccinated quickly and in many places will be a challenge, and that's scaling up to involve not only the states and territories Pfizer hubs, but other clinics in various places, particularly in remote and rural areas over the coming weeks and months, through general practices and into pharmacies as we go into stage 2. And the final thing I'd say is keeping the confidence in the vaccination program. That's going to be absolutely crucial to make sure that vaccine hesitancy is removed as much as we can, and to make sure people know that these are safe, effective, and high quality vaccines, and that this is the way out of this pandemic to an extent, but certainly to save lives is the crucial thing. And a recent survey here in the ACT, which was shared with me today, showed that 60% of the population would get the vaccine immediately if they was offered. The 10% don't want it at all, and 30% are not sure. And that not sure area is the one that's been rising in recent weeks. And I'm really confident and hopeful that as the vaccine rolls out, that number will decrease. But I think it's for all of us on this call, it's really important that we do all contribute to that confidence piece, because that's the way we're going to succeed in this extraordinary effort. That's the way we're going to protect lives, particularly those that are most vulnerable, and our ability to start to work towards some sort of post-COVID normal in the months ahead, which I'm sure we're all looking forward to. So Christine, I'll leave it there. I'll be very happy to take questions during the panel, but I'm looking forward to the other speakers. Thank you.
Oh, thank you very much, Paul. And I think that's an excellent summary, and it's very salient, important points as we're on the eve of starting the program on Monday. So it's my great pleasure, I've already introduced, but I think now to hand over to Professor Andrew Pollard, Professor of Paediatric Infection and Immunity at the Oxford Vaccine Group, University of Oxford, and the Chief Investigator leading the development of the Oxford AstraZeneca vaccine. So Andy, thank you so much for joining us early in the UK with the coffee on board, and we are really looking forward to what you're going to share with us this evening here in Australia.
Thanks very much, Christine, and great to be with you all today. Let me just get my slides up. Great, hopefully that's projecting now. Okay, so I'm going to talk a little bit about the development of the vaccine here from Oxford, and about the global trials. And I mean, it's interesting talking to you in Australia, that if we think back to just over a year ago when this all started, the news we were hearing here was about the bushfires in Australia reaching Brazil, and certainly at that time I didn't expect to be spending the year working with close colleagues and friends in Brazil. And just a few days later, in early January, there was this article in the New York Times which discussed an outbreak of coronavirus infection in Wuhan, but was very reassuring, because it says that there's no evidence that this virus can spread between humans, and how wrong it turns out that that view was when we look back at what's happened in the world over the past 12 months. The UK had the first cases of the virus just a month after that. We had 3 people positive by the 6th of February. And we had, by then, had already got underway with thinking about how to move forward with vaccine development, and had constructs which were going into manufacturing. And on the 11th of February, we got together, myself and colleagues from some of the other vaccine teams in Oxford, to start discussing how we would put together a clinical development program, given the huge efforts, particularly by my colleague Sarah Gilbert, in making the first vaccine constructs during January. And so we began our planning at that stage to move as fast as we could, as soon as we had product that had been through GMP production. And as you heard in the introduction, the world now looks very different, with this huge impact on humanity as a result of the pandemic during the past year, with some millions of deaths and hundreds of millions of cases. And as I think was just outlined in the introductory comments, that really the focus of vaccine protection has to be in those at the greatest risk of disease, and that is older adults and those with other comorbidities. And my teenage son was very pleased to point out that I was 100 times more likely to die from coronavirus than him. Now, the virus is decorated on its surface by spike protein, which binds to the ACE2 receptor. And as I mentioned, the genome was available in January, which allowed the vaccine developers around the world to get started on vaccine planning, design, and to make constructs. And it is worth just reflecting what an amazing advance this is over the last few decades, that within a matter of weeks of the virus being recognized, that we can have global distribution of the critical piece of information needed to develop vaccines. And of course, the other thing which has really meant rapid progress here is that we've had these 2 previous outbreaks of coronavirus infections. So the coronavirus had already reached the list of viruses that had pandemic spread potential. The first on the left of this slide, the SARS outbreak about 18 years ago, that had about 11% mortality, but fortunately didn't transmit very well and was contained by isolation of cases. And then more recently, the MERS outbreak, very scary, 35% mortality, and again was contained by case isolation. And we were fortunate with both of these viruses that they didn't transmit between humans particularly well, and but they did drive a lot of efforts in vaccine development, which meant that we've understood a lot about the biology of coronaviruses, and also the key antigenic sites on the virus that are important for the development of immunity. And by those observations and animal studies with both of the SARS and MERS virus, it was known even by the beginning of this year, the beginning of last year, the right direction of travel to make vaccines for coronavirus. And Sarah Gilbert had already started clinical trials of a MERS coronavirus vaccine that had been underway both in the UK and in Saudi Arabia, which meant that we had a lot of experience of working with our vaccine construct for a coronavirus before the outbreak began. Now, those of you who work in vaccine development will know that it's a long process, and you have to have pretty good perseverance to get from the design end right through to licensure. And most of the vaccines I've worked on have taken at least 10 years from the initial design through to deployment, and often longer than that. And that's because we run the processes in this way in series, not in parallel, and usually have gaps for between each of the steps to acquire funding, to get through complex approval processes and bureaucracy, and also often to wait for slots of manufacturing to actually make the vaccine for the different phases of development. And then even when you get to the completion of phase 3 trials, the process of regulatory approval can be very delayed. And in the context of the pandemic, many of these steps have been shortened by dealing with the bureaucracy, making sure funding was available. And I think critically, the manufacturing of product at great financial risk to make sure that when the clinical trials were moving, and then finally for production for deployment, that vaccine doses were available. And this is an extraordinary situation for those of us who have worked in development for a while, and that these components have been put in place. Having said that, I have absolute confidence that there have been no corners cut through any of these processes, because the degree of scrutiny through our trials that we've been under from regulators and from ethical oversight and the manufacturing side is exactly the same as would happen in peacetime. And of course, the other point is that the scale of the trials that have been undertaken for all of the vaccines in development are simply enormous, which I think also gives us great confidence that we have huge safety databases for each of the products. As many of you know, there's somewhere between 200 and 300 vaccines currently in development, and a large number of those already in clinical trials, and some, as we were hearing, already licensed. And they cover a whole variety of different technologies. But apart from the inactivated whole virus vaccines, the vast majority are focusing on this surface protein, the spike protein. And that is, I think, an important feature, which means that we have quite a lot of confidence, even between the platforms, about the performance of the different vaccines. And that's because, to a large extent, all of these vaccines are essentially protein vaccines, whether they start as a viral vector like ours, or as DNA, or as an RNA vaccine like the Pfizer vaccine, or one of the protein vaccines. In the end, they all result in the production of protein for recognition by the immune system to make an immune response. With the viral vector vaccines, they contain spike protein DNA, and after immunization, our cells are then harnessed to unpack the DNA, convert it to RNA, and then into spike protein to generate an immune response. And there will be some differences between the vaccines because of different expression levels of protein, and because of the danger signals that the different products present to the immune system to soup up the immune responses. But overall, they are all generating both antibody and T-cell responses against the spike protein. So the vaccine that we've developed here is based on a chimpanzee adenovirus. And on the right of the screen, you can see the adenovirus. It's non-replicating, so it cannot cause ongoing infection in humans. Even in the immunosuppressed individual, it's perfectly safe. And that is as a result of the deletion of some critical genes from the virus, which means that once it's unpacked its DNA, is not able to go on and replicate further. And into the genome of the chimpanzee adenovirus, the spike protein gene has been inserted, so that after immunization, the spike protein gene can be expressed in our cells. So we started our phase 1 trials in April last year with a plan to look at safety and immunogenicity with a cohort of 1,000 younger adults in the UK who were healthy, and watched them very carefully. And from those trials, we found that we got very good responses to the first dose. This is neutralizing antibody response on this slide. And in a small subgroup where we looked at 2 doses, we saw a boost in those antibody responses. The initial plans for development had been as a single dose vaccine, but with these promising results, which we then confirmed with further booster doses in another subgroup, we decided to switch to a 2-dose program because of the enhanced immunogenicity of the second dose. And we also, in this study, observed good T-cell responses, which you see on the right of the screen. And that switch to a 2-dose program had some interesting consequences. And the main one of those was that there was some delay in the availability of the second doses within our trials, which meant that when we finally managed to complete the phase 3 trial, we had a varying dose interval from 4 weeks to 12 weeks, which has demonstrated this interesting phenomenon that there are better immune responses with the longer dose interval. The phase 2 trials in older adults, again, healthy older adults, started in May of last year. And these were important because they showed here again on neutralizing antibody assays that whatever the age group, the immune responses were very similar. In green are younger adults, purple is 56 to 69-year-olds, and the gold color are those over 70. And so after 2 doses, all age groups had strong immune responses, which were very similar. And this slide also confirms that in older adults, that a single dose is less immunogenic. These are the 2 lines at the bottom here, and also that there's good persistence, at least up to day 56 with the 2-dose schedule. We moved very rapidly in June to phase 3 trials. And in these trials, we had groups in the UK, Brazil, and South Africa. And you can see here in total, there's around 24,000 individuals, and around 20% are over 55 years of age. So these are the trial sites, 19 across the UK, 6 in Brazil, and 6 in South Africa. We also have a small trial ongoing in Kenya. And each of these sites were chosen as we were very keen to have global representation in our clinical trial portfolio, because right from the beginning, our plan was to develop a vaccine that could be made available throughout the world, and particularly for those who are less able to access vaccines normally, and to try and make sure that we were developing a vaccine which could be widely distributed. And part of that was a vision from the university for a not-for-profit development program. And we were fortunate to find a partnership with AstraZeneca who shared that vision of not-for-profit, which is unusual with pharma generally. And I think that has been really important in the progress that's been made since then. They have a trial in the United States of 30,000 individuals, bringing our total trial population to around 55,000 individuals. In Russia, there's a small immunogenicity trial, and also in Japan. And a third partnership with the Serum Institute of India, which has resulted in some trials in India, and also the vaccine being manufactured on huge scale to provide availability to many other parts of the world. So with those phase 3 trials, we rapidly managed by November to get to a point where we could have an interim analysis, and did a database lock on the 21st of November. And by that stage in the trials that I've been running, we had 23,000 volunteers enrolled. We'd taken over 100,000 blood samples, and had 500,000 pages of data that needed to be cleaned and prepared for analysis. And the headline news was, with that initial analysis, with the analysis that was pre-specified with regulators, had a 70% efficacy. And importantly, in all those who were vaccinated, there were no hospitalizations, severe cases, or deaths. I'll come back in more detail to the efficacy analysis in coming slides. And the interim analysis led to regulatory approval of the vaccine at the end of December here in the UK, and the rollout of the vaccine that began on the 4th of January. The regulatory status now has expanded. There's around 50 countries that have approved the vaccine, and just over the last week, as was mentioned earlier, TGA in Australia, but also I think importantly for our global mission was the World Health Organization policy advice last week, and then at the beginning of this week, the approval by the WHO pre-qualification team, both of the AstraZeneca version of the vaccine manufactured in Korea and the Serum Institute of India version of the vaccine manufactured in India, that provides the global approvals for rollout of the vaccine through the COVAX mechanism, which I think is perhaps the biggest landmark for us in the last 12 months. There's also a large effort to make sure that the vaccine supply is coordinated in a way that allows the vaccine to be protected in its supply chains in the various different geographic regions of the world. And here you can see manufacturing sites and the provision of vaccine to different regions that has been coordinated by AstraZeneca to ensure that a global access plan is achieved. So we've updated our analysis with a further data cut in December that provides additional information, and I'll just take you through a few of the headlines from that analysis that may be helpful. And the first is around the protection against hospitalization and severe disease. And what you see on this slide is that the hospitalizations, as 22 in the control group during the course of the trial up to the 7th of December, and only 2 in the vaccinated group. And of those 2 in the vaccinated group, one was an individual who was hospitalized on the day of vaccination with COVID, and the other is an individual who was hospitalized 10 days after vaccination with COVID. So both of those could not be protected by the vaccine. But amongst the overall picture, once an immune response to the vaccine has been established, there are no hospitalizations or deaths caused by COVID, I should say, in the vaccinated group. And this has been, I think, an important finding for all of the vaccines in development, this very high protection against lower respiratory infection. So one of the big questions that was asked from a policy perspective is, what is the protection from the first dose? So if you give the first dose, how good is the protection? How long does that last? And we measure protection from 3 weeks after the first dose. The immune responses are strong by 2 weeks, but in order to wait for a washout period of infections acquired before 2 weeks, we start counting from 3 weeks after the first dose. And what we find is that if you follow individuals from the first dose up to 3 months, protection is around 76%, and it doesn't wane at all in that period. We see very similar levels of protection from the first dose right up until the second dose is given at 3 months. And this is also mirrored by fairly persistent antibody levels over that period. The purple line is the dosing regime that's used. So this is just after a single dose up to the 90 days. There's almost no change in antibody level. Even if you go out to 182 days, and there's very little change in the immune response over that period. So that gave confidence here in the UK for an extended dose interval with good protection after the first dose, persisting right up until the second dose is given. And then if we look at the effect of that interval on the efficacy after the second dose, you can see here that there's higher efficacy with the second dose given at 12 weeks or more after the first dose, and a rise in efficacy over the course of that if longer dosing intervals are selected. So that's justified both the extended dose interval and the improved efficacy after the second dose, which is seen here that as you extend the interval, there's a gradual rise in efficacy over time between the first and the second dose. Another really important question which we're asked about is quite confusing to understand is about transmission. And I think uniquely in the UK trial, we have taken swabs from everyone in the trial every week. So that's hundreds of thousands of swabs since the trial started last June. And we're therefore able to get a picture amongst our trial population of the extent to which individuals are PCR positive during the course of the trial. And I'm going to just talk for now just about that initial period after the first dose. But if you imagine that amongst your population, there is going to be a group of people who are hospitalized, some who have mild symptoms, some who are asymptomatic, and some who are not infected. If the vaccine stops hospitalization, reduces symptomatic infection, but has no impact at all on PCR positivity, you would expect no change in the number of people in the population who are PCR positive. But what we see is around about a 2/3 reduction in PCR positivity amongst those who are vaccinated, which I'll show you on the next slide. And it's important to remember that around 1/3 of cases are asymptomatic, but 2/3 of cases are symptomatic. And so overall, if you look during the course of the trial, you can estimate the amount of reduction in transmission that you should have if you assume that a negative PCR means you're highly unlikely to transmit. And so what you can see here is that after the first dose, we see a 100% reduction in hospitalization, 76% reduction in mildly symptomatic infection. And although there is little change in the asymptomatic infection, it hasn't gone up, which, as I said, you might predict that it would if there was no impact on asymptomatic infection. But indeed, in those who are still PCR positive, they have a lower viral load and a shorter duration of shedding. And so we've got around about a 2/3 reduction in PCR positivity in the population. And even the third who are still positive are likely to, or less likely to transmit because of that lower viral load and shorter duration of shedding. And so that, I think, suggests that in the context, at least, of the clinical trial, that one would expect the vaccine to have an impact beyond protection to actually reduce the number of people who are infected and able to transmit in the population. So next, I just want to talk a little bit about the UK variant known as B.1.1.7, which spread very rapidly from the end of last year to all corners of the UK and has now spread to many other countries too. And this variant arose to become the dominant variant in the UK from about November, but really within the trial, we didn't see very many sequences until well into December. And so the data here are not from a formal database lock, but they come from the swabs that we've been able to sequence up until this point. And the first thing to say is that in the Public Health England assay, we do see, compared with our prototype strain from Victoria, which is close to the Wuhan variant, that we do see a reduction with the UK variant in neutralizing antibody activity. And you can see that shown on this slide. But interestingly, we see relatively similar vaccine efficacy. The efficacy of against B117 is 74%, and the non-B117 variants is 84% in this analysis. And these are similar numbers with overlapping confidence intervals, but clearly this doesn't exclude the possibility that there is a slight reduction in the efficacy against B117. But overall, we still have high efficacy documented against this new variant. And interestingly, that's despite that reduction in neutralizing antibody, which obviously then raises questions about the neutralizing antibody and how well that relates to a correlation with protection. So I'm going to stop there at this point and just say that we see continued high protection against hospitalization and severe disease, which is clearly the most important feature here. We're not very worried about mild upper respiratory tract infections caused by coronavirus. The thing that matters to our health systems is the severe disease. That we've documented in the UK good protection from one dose for the 3-month interval, a really good boost both on the antibody side as well as in efficacy with the delayed dose interval. And protection against the UK variant. We have a large study in Brazil, as I mentioned earlier, and we're just sequencing the variants or the swabs there at the moment, and hopefully we'll have more of an idea about the circulation of strains in the trial in Brazil that will help allow us to do a similar analysis about protection against the P1 variant that has emerged in Brazil. In the studies in South Africa, we undertook a small trial of which was phase 1/2, focused on young adults who, as you know, only develop mild disease. And as you would anticipate, we saw very poor protection against mild disease because the B1351 variant that arose in South Africa has developed to evade replication, or at least inhibition of replication by the immune system in the upper respiratory tract. And we know that that's an expected finding because in individuals who were infected in the first wave in South Africa are just as likely to be infected in the second wave as individuals who are naive to the virus. So the virus has emerged with these new variants to avoid human immunity in the upper respiratory tract. But I think reassuringly, a very similar vaccine from Johnson & Johnson, where they did look specifically at severe disease in their population, so they vaccinated older adults, they saw good protection against severe disease. Similarly, with the Novavax trial in South Africa, a very similar story. And that, I think, may well suggest that the vaccines are able to prevent hospitalization and severe disease because the mechanisms of immunity, such as T-cell responses, are not compromised by the virus's need to mutate to allow it to continue to transmit and to survive in human populations. And I think that probably is an important message for us for the future, that this virus, as with all viruses that we live with, is going to have to continue to mutate in the future to persist in human populations, and we're going to have to learn to live with it. And that might be the current generation of vaccines are enough to prevent hospitalization and severe disease, or we might need to update vaccines to maintain protection, particularly in the most vulnerable. And of course, all developers are working hard to make sure we're in the best possible position to manage whatever the virus throws at us next. And I'll just end there. Thanks to the large group of volunteers who've taken part in the trials, the team here in Oxford, as well as our many investigators in South Africa, Brazil and the UK have worked tirelessly over the last year. Thanks very much.
Thank you, Andy. That was absolutely incredible, a tour de force, and I think an amazing snapshot of what has been a global mission, which, you know, the world owes you and your team a debt of gratitude. So I will hand to Katie Flanagan to thank you more formally in a minute, but I'm going to just briefly introduce Hao Cheng and Dr. Hao Cheng and Associate Professor Michael Nissen from the TGA to give us a very quick refresher update on the pharmacovigilance plans for Australia as we start the program rollout from Monday. So over to you, Hao and Michael.
Okay, thanks very much, Christine, and appreciate the opportunity just to really quickly introduce the TGA and the Australian Government Safety Monitoring Plan. I just wanted to really just tell you that the vaccine surveillance section within the TGA is specifically set up to monitor the safety of vaccines that are used in the Australian population, and it's a multidisciplinary team of doctors, public health physicians, pharmacists, nurses, biostatisticians, epidemiologists, who are looking at all aspects of AFI or adverse event reporting that comes to us from the states who are the main immunisers. So in the interest of time, I will just now pass on to Dr. Hao Cheng, who will give you an overview of the safety plan that will be rolled out with the vaccines. Thanks, Hao.
Hi, thanks, Michael, and good evening. Just check that there's sound. You can hear me?
Yes, thanks, Hao.
Thank you. So in the interest of time, and I realise a number of you have attended last week, I'm going to skip a large number of slides and head to some of the key things. So if we go to slide 4, please. As part of the National Surveillance Safety Surveillance System, we have, sorry, it's next one again, 5 perhaps. We've developed we've developed a number of enhancements to the existing National Safety Surveillance System at the TGA. And the key objectives in doing that is the timely collection of adverse events following immunisation and the management of those through signal detection and investigation and actions to address any safety issues that come up from reporting. Go to slide 12, please. So in order to support this effort, every single report that you may generate is valuable because it helps us to build a better picture of the safety profiles of these vaccines in Australia in the real world setting. There's many ways that reports can come to us through the TGA, and in fact, most roads lead to the TGA regardless of where you start. So consumers can report through to their healthcare professionals, and thank you healthcare professionals for taking those reports seriously and reporting to government. Consumers can also report through to the National Prescribing Service, the NPS, and there's a phone line on their website where they can sit down with a pharmacist, and a pharmacist can guide them over the phone about those adverse events, generating a report that goes straight to the TGA. In terms of healthcare professionals, just again to emphasise that adverse events following immunisation with COVID and other vaccines are notifiable in New South Wales, Western Australia, Queensland, Northern Territory and the ACT. The reports given to the states and territories are then passed on to the TGA, and we generally prefer that you do report straight to your state or territory health department because of those notifiable requirements for a number of states and territories. Reports can be sent directly to the TGA, and there's a website that explains how to do that, and also the links to all of those other state and territory and NPS contacts as well. I want to talk for a moment, I don't have a slide on it, so just pause on that slide, on what to report. So certainly we're very interested in any medically significant events that occur following immunisation that might be related to immunisation. And in particular, those resulting in serious outcomes. We'll take your clinical judgment as to what you think a serious outcome is. But in particular, and not excluding others, certainly if there's an outcome that seems life-threatening, that requires hospitalisation, that results in prolonged hospitalisation of existing hospitalisation, results in significant disability or incapacity, results in congenital anomalies or birth defects, or even if there's a suspicion that it may have contributed to a death. Also, any adverse event that requires intervention to be able to prevent any of these sorts of serious things. So even if you're able to prevent something serious, if you think something serious might have happened, it might be good to report. A couple of other types of things to report include adverse events where the event seems unexpected, something strange going on. I would not have expected that to happen, but it's happened, so I might report that. Also, if there's any concerns about the use of the vaccine vials themselves, using multi-dose vials in Australia is something that not a lot of people are used to. And so if there are any concerns about the use of these multi-dose vials, certainly report those where it relates to a specific encounter with a patient. Apart from that, we do encourage you to report where you think the TGA should know about something. We're certainly keeping our eye on a number of things behind the scenes. But if there's something that you suspect, we think the TGA might want to know about that, please report. Also, if you're not sure, you can also err on the side of reporting, because the more information we get on these vaccines, the more we'll be able to pick up any safety issues, should there be any unexpected things. But also, importantly, the more we're able to reassure the Australian public where in fact there aren't a lot of things going wrong with these vaccines. And that's a really important role in building vaccine confidence. So we encourage you to report, and thank you for the opportunity for passing on that message, Christine.
Sorry, thank you very much, Hao, and I'll put in a shameless plug as well for the fact that NCIRS is leading the AUSVAC safety program, which will of course be conducting active surveillance through many of the sites where COVID vaccines will be delivered. It's my pleasure to introduce Katie Flanagan now. Over to you, Katie.
Thank you, Christine, and thank you for inviting me to be part of this Q&A session. So I'd just like to start by thanking, actually, Professor Kelly and Professor Pollard for coming and giving these wonderful talks today and giving us some insights into the program here and into the Oxford AstraZeneca vaccine. Just to begin with, I'm going to ask Professor Alan Cheng to come on and just give us a few words about the clinical guidance that will be released soon for the Pfizer vaccine. We've already had the clinical guidance from ATAGI released for the AstraZeneca Oxford vaccine. So we've now got clinical guidance that combines both of these vaccines. So over to Alan just to give us a few words about that.
Yep, so I've been typing away at the Q&As, which get longer than I can respond to them. But I think a couple of those answers have the links to the Department of Health website, which is getting a increasing number of documents that will be helpful for clinicians. And there's certainly information for clinicians for clinical guidance in general, and then specific ones for AstraZeneca and for Pfizer vaccines. So probably not much more I can say about that, except to say that it's coming and will be available well ahead of when the rollout will be.
Thank you, Alan. Right, so we'll start with some questions. So maybe if Paul Kelly and Andrew Pollard would like to put their screens back on, and we can ask a few questions. So I'm going to start with one for you, Andrew. The low dose, high dose issue. Can you just give us a little bit of insight into that? Because I think this is a strategy that's not being pursued. Everybody was talking about that higher efficacy, but many of us have seen the data. It looks like there was longer time intervals. So could you just give us some insights into that?
Yeah, absolutely. I mean, I think the most likely explanation for the higher efficacy is because there was a longer dose interval in that group. And so the main reason why it's not pursued is that we actually have the largest body of data around the 2 standard doses. So from a regulatory perspective, we don't really have enough data to provide the sort of the robustness of support around the low dose standard dose group. And since it now looks, once we had the immunological data, as if it's largely driven by the dose interval and the higher antibody responses that you see with a longer dose interval, which match, in fact, a longer dose interval with the licensed product, it doesn't seem necessary to have a complicated system of having a half dose followed by a full dose. I mean, there clearly is one advantage in doing that, in that you've got more vaccine doses to spare, but it just is much more complicated to deliver operationally.
Okay, thank you. I'm going to hand to Kitaki now. We're taking it in turns to ask questions and try and work our way through the numerous questions that we've got here.
Thanks, Katie. Yeah, we've been watching the Q&A closely and there are so many amazing questions, so I'll speak quickly. This one is also for Andrew, given that we know you have to leave a little bit early tonight. The most popular question for you, I think, is how do you explain the efficacy of above 70% for a single dose versus around 62% for 2 doses with a 4-week interval? And that was in the Lancet preprint post hoc analysis.
Yeah, I mean, I think the issue is to do with the complexities of the populations that you're looking at different times. And so you know, when you're looking at between the 2 doses, it's a particular time in the pandemic and different combinations of individuals that are available to include in that analysis. So different proportions from different countries, different age groups and so on. And compared with what you look at in the post 2 doses, which then is a group that is involved, for example, in much later with the next wave of disease and so on. So it's very difficult when you're looking at different populations that can be included in an analysis to get exactly the same number. The reality, of course, is that there are overlapping confidence intervals between those 2 numbers anyway. So it's difficult to know whether they're true differences. But you know, I think overall it's pretty similar in terms of the efficacy after 1 or 2 doses, but it's higher if you push out to 3 months and have a stronger booster response. I think the other thing that's important is that there does seem to be some increase in efficacy with that dosing interval. And you know, there is another interesting question about whether it would be higher if you waited even longer. But clearly, from a practical perspective, going for longer it doesn't make sense.
So now back to me, and I'll ask Andrew another question. How confident are you that your vaccine is efficacious in the over 65-year age group, given that there isn't a huge amount of data?
Well, I'm confident based on the immunological principles. We've got very similar immune responses in older adults as in younger adults. The efficacy we do see is in the same direction. It's just smaller numbers, which makes it less robust. But the reason why it's smaller numbers is because we started the older adult trial later, so we've got less time to accumulate data in the older adults. The overall sample size is a bit smaller. It's because it's 20% of the population. And then lastly, older adults are perhaps much more conservative about their interactions with other people during the pandemic, so they have less opportunity to meet the virus, which means that you have a lower attack rate. So all of those things put together means that you need more time to accumulate the data, and we're rapidly getting to that point. But of course, here in the UK, the real world evidence is going to be on an enormous scale when that reports, hopefully in the not too distant future. So I should also say the US trial has 24 or 25% over 65. So we're going to have a lot more data in the very near future. So I think at the moment, based on the principles of the immunology and the efficacy signals, it looks as if it will be very similar to younger adults, but we'll know more in the very near future.
Alan, you want to chime in?
Yeah, I was just wondering if I could ask a question. There's a lot being made about, the headline results from different trials, but they're not head-to-head trials, and there are differences in the study protocols. And I know, for example, looking at your study protocol, there were weekly reminders, and if someone had a symptom, you sent someone out to them to get a swab. in other trials, it was, there was self-swabbing and then the swab was sent into a central lab. Do you think those differences can make much of a difference to efficacy estimates?
Well, I think there's a lot of different factors that will affect efficacy estimates. So the population that you're working in makes a big difference. If you focus in older adults, there's a lot more severe disease, and it's absolutely clear from all of the studies it's easier to prevent severe disease. So if your population has severe disease, you will by definition have higher efficacy. Different populations and different. I think the point you make really about the different ways in which you acquire the patient to do the swab will make a huge difference to the efficacy. So I think you're right. You can't compare head to head of the different trials at this moment, but that doesn't exclude the possibility of real differences in efficacy. But I think at the moment we can't make robust estimates of what the difference would be between the vaccines. I suspect we'll get more data from, as I say, from real world evidence on that. The important thing in my mind, though, is the reason why we're vaccinating people is not to prevent mild infection, it's to prevent hospitalization, severe disease and death. And consistently, whichever vaccine you look at, there is very high efficacy against those. And that to me is the hardest endpoint, even though there's differences in hospitalization, utilization in different trials, we're still seeing, when you go to that more severe endpoint, almost identical numbers, whatever vaccine is used.
Thanks, Andrew. I have a question now for Paul, please, which is, as a nation, we're so lucky that we have already secured agreements for more than enough doses for our entire population. So are you able to tell us a little bit about the plan to support countries in our region, and whether, could you give us a number of the number of doses that will be offered as bilateral donations?
So I can't give specific numbers, but I'll make a couple of comments. Thanks for the question. So the first thing is we were one of the first countries in the world to sign up for the COVAX initiative, and Andy, Professor Pollard mentioned that initiative, which is a wonderful thing in terms of making sure that we're all contributing where we can to increase the possibilities that particularly low and middle income countries can afford and be provided with vaccination. So we did that early, and we're certainly remaining a strong commitment to COVAX. In our region, there have been bilateral arrangements with many of the Pacific Island countries and territories, as well as several countries in Southeast Asia, in relation not only to the provision of the vaccine, but also support for the vaccination program. And finally, if you add up all of the doses that we have pre-purchased for Australia, it's highly unlikely that we'll be vaccinating our country, everyone in our country 5 times. So we will have vaccine to share with the region too. So that will play out over the year in terms of, particularly as Professor Pollard has talked about, how the vaccination may roll out in elderly people as well as older Australians. I get caught up for saying elderly, older Australians, as well as what might happen with the variants of concern. But there'll be plenty of vaccine to share.
Okay, I'm going to go back to Andrew again for another question. So the TGA have approved the Oxford AstraZeneca vaccine for 4 to 12 weeks. That's the indication. Now, looking at your data, obviously this is a post hoc analysis, but it looks pretty convincing that that later time point is more sensible. So should we be preferentially saying 12 weeks?
Well, I mean, I think just on the normal vaccine principles, we know longer gaps between doses tend to give better booster responses and give time for B cell maturation. So I think having longer dose intervals is in some ways not surprising because we've established that previously with protein vaccines and so on. But I think that the decision making on exactly what intervals are chosen for Australia is up to the local policymakers to decide.
Thank you. And I think Christine has her hand up.
Yes, so it's actually, I won't preempt what will be coming out in the clinical advice around AstraZeneca, but I'll leave that to Alan if he wants to comment. Probably shouldn't, but Andy, I know I'm conscious that you're going to have to leave us very soon, and I wanted to ask you the crystal ball question that I think we were talking about when you first signed on this evening. And it was really, where are we going to be with these variants in 6 or 12 months' time? Will we be seeing, SARS-CoV-2 as a virus that causes mild disease and we're preventing it severe disease with our vaccines? What's your opinion on the next 6, 12, perhaps 18 months?
Well, I think it's highly likely that that this virus we will live with in the decades ahead. It's not going to go away. It's not going to be eradicated from the planet, unfortunately. And so the question is, what does living with it look like? With other coronaviruses that are very common childhood infections, they cause mild illness in childhood, and then as adults, we have upper respiratory infections every few years with them, and we shake them off. Now, it would be great if we get to the point with this virus where this is what we're living with. And I think the open question at the moment is, does the current generation of vaccines convert our immunity into a situation where, at least if you're vaccinated, we can just live with the virus and have mild infections and we forget about it in the future? Or is it going to be more like influenza, where as new variants arise, we have to update the vaccine perhaps every year to make sure we stay on top of it? I don't think we can fully answer the question at the moment, but there are some signs in my mind that give me some optimism based on the trials I mentioned from Johnson & Johnson and Novavax in South Africa, which suggests against that really important hard endpoint of severe disease and hospitalization, that the current generation of vaccines still have high efficacy against those endpoints, even though we're dealing with a variant that will evade the replication in the upper airway and likely mild disease. So I think we have to be prepared. And so that's why, as I mentioned, all developers are working on new generation of vaccines for the new variants. But I think the jury's out at the moment about whether we really are going to need them. So that's my optimistic view, that maybe we're a long way forwards already. But we have to continue to monitor carefully and be prepared.
Thanks, Andrew. And I hope this doesn't feel like an interrogation, but I know you have to leave. So one last question from me for you, which has come up in the pre-seminar questions as well as on the Q&A. Very simple. Does the vaccine contain DNA or mRNA? And then is there any theoretical concern about integration into the host genome if it's DNA?
So there's no evidence of any integration into the host genome with any viral vectors or any problem with persistence with the RNA vaccines either. These are non-issues. Adenoviruses are DNA viruses. So the spike protein gene is a DNA sequence within the adenovirus, but it then gets converted into RNA and then into protein. And obviously the RNA vaccines are messenger RNA, which is then just converted into protein. So in many ways, as I said at the beginning, they're sort of all dressed up protein vaccines. They've got fancy names like being viral vectors or RNA vaccines, but in the end, they're protein vaccines. And the differences are much more around the danger signals that they're posing to the immune system to help make strong immune responses.
So Andrew, do you have to leave now, or can I ask just one question with that in mind, which is about...
One last question, I really do have
One last question, yeah. Anti-vector immunity, I've heard that asked many times. Have you got any evidence at the moment? Because obviously we'll be thinking about boosters down the line.
There's two sort of lines of evidence. One is that from the trials that we've done so far, there is some anti-vector immunity generated by the first dose. But it doesn't seem to compromise the response to the second dose. That's what we've looked at, are neutralizing antibodies against the adenovirus. And the level of those neutralizing antibodies doesn't seem to correlate with any reduction in spike protein response to the second dose. So that's reassuring. We also have a small subgroup in the trials who had previously received the adenovirus in trials of other inserts in the last few years before the pandemic. And those individuals also make good responses to 2 doses that are very similar to naive individuals. But it does need studying in more detail for boosters, and we do have plans to answer that question. If you've already had 2 doses last year, what happens if you get another booster this year? So we'll look at it formally, but so far the data look quite encouraging.
Thank you. So I think you need to leave, and I'm sorry we've really hammered you with questions there, but it was irresistible having you here and being able to ask some of these questions to somebody who really knows the answers, because we've had many debates at ATAGI around lots of different things. So we're very, very grateful that you gave up your time today, and thank you so very much.
Pleasure. Always happy to be interrogated by Australians.
I'm British.
Okay.
Thank you.
Bye-bye. Thank you.
So I think now we can turn our focus to the neglected members of the panel. I'm sorry. So my next question will be for Paul, please. There's a couple of questions in the Q&A about stage 1B. So people in the general public who have chronic medical conditions or disability, how will they actually be identified and offered vaccination?
That's a very good question. I don't know if Alan may be the best to answer that. Sorry to put you in it, Alan, but I know that ATAGI's turned their mind to this recently. So first, I will say a couple of things though. So firstly, there's a number of questions, and I've tried to answer a few of them about GPs having access to vaccine for their own patients. Obviously, for GPs that have applied in the expression of interest round to be part of this, they will be rolled out during 1B. We'll be studying the numbers of GPs, but it's an active discussion about how quickly we'll program. So of course, if you're going to your regular GP, they will know who those people are. Second of all, one of the easiest parts of that 1B process is the age cutoff. So having that proof of age is an important component for wherever you end up, in a state clinic or one of the GP respiratory clinics or your regular GP. But in terms of the chronic diseases which are there in 1B because of their increase in severe risk of severe COVID infection, Alan, have you got anything you'd like to say there?
Oh, look, I mean, I think, ultimately we want everyone to be vaccinated. So, I mean, I think a bit of clinical judgment needs to come in, and, I mean, there is a list in the information as to what, constitutes a chronic disease. But, if someone is, coming to you, you've seen them before, they've got asthma, I'd just go ahead and give it to them if they qualify for it.
I think I'd just add one other thing is that, the reason why we have this priority grouping, I mean, because as I said, we're trying to save lives and give people, the vulnerable people, the vaccine as quickly as possible and hopefully before winter kicks in. But the biggest issue, if we had enough supply today to offer it to everyone like we normally do in the National Immunisation Program, we probably wouldn't have a prioritisation. But the reality is, at least for the short term, we do have a limited supply of the Pfizer vaccine, and that will continue actually right throughout the year. We're not getting 20 million doses all at once. There will be regular deliveries through the year. AstraZeneca at the moment, we don't have any, but that will turn around rapidly. So as Alan said, by the time we get into April, we will have millions of doses of AstraZeneca, Oxford AstraZeneca vaccine. And so we will go along those priority groups, but we'll be quickly going into the other phases of the program when that supply issue is no longer a major issue.
And probably one thing just to add to that is that the phases aren't like you finish phase 1A and then you enter phase 1B. They sort of, not all the people in 1A are going to be vaccinated by the time phase 1B starts. So they sort of just, it's just, the idea is to give some of these more vulnerable groups a bit of a head start to get some vaccine, but they will sort of flow into each other.
So a question for Alan then. This has been asked by a lot of people. It's about safety with either of the vaccines in immunocompromised people, people on chemotherapy, people on immune checkpoint inhibitors. Could you just give your comments on that?
Yeah, I guess there's sort of two parts to that. And so the first one is about safety and the second is about efficacy. So obviously with immunosuppressed people, there is a concern that efficacy may not be as good, but obviously the other side of that is that, vulnerability to the disease is more, so they probably balance themselves out to some extent. In terms of safety, I think that's a bit more of a difficult issue. I think, there are diseases where I guess we're not quite so concerned about having an inflammatory component to things. So, HIV and some of the immunosuppression syndromes. But I think there was a question on this about, organ transplantation and people, on checkpoint inhibitors where there is some concern about, triggering off autoimmune phenomenon and so on. I think in that group, it really is going to have to be a clinical discussion that weighs up the risks, and it's not really the harms; it's the lack of the lack of information about harms, so potential harms, and so it really depends on who that person is, what they decide, what their level of risk is, if they're someone that is likely to be highly exposed to or is immunosuppressed to the extent that if they got COVID they would run into trouble, then that would be a discussion that you might say, well, on balance it's probably better to get vaccinated. I guess the other thing is that as the 130 million doses that have been given so far, there will be data emerging soon on the safety in particularly in autoimmune and inflammatory diseases. So you may want to just wait a little bit just for some of that data to come out. But in general, I probably wouldn't have too many concerns about these concerns have been raised with flu vaccines and other things before, and in general haven't shown to be a huge problem. So I think it is one for a clinical discussion.
Alan, I have a question for Michael or Hal now, please, which is, do you want people to be reporting every mild or moderate expected side effect, or do you have particular criteria for which adverse events should be reported after vaccination?
It's Michael, and I'll also allow Hal to comment, but our wish is actually send us all the AFIs that you think, as Hal very nicely presented, we're interested in all AFIs. And, this is a new vaccine program that we're rolling out. So we really want to have high awareness of what its safety profile is in the Australian population. And also that then underpins vaccine confidence in our country. So please send us the reports. But how?
Thanks, Michael. Yeah, and I think one of the key perspectives on the milder reactions is whether this is unexpected. So some mild reactions or mild symptoms may actually be a symptom of something quite serious that might take some time to occur. So I think the key thing is whether it's unexpected, either in the nature of what happens, or if there's a sudden spike. If you see a run of a lot of mild things going on, then that seems really unexpected. We'd like to know about that, because then we can say to the Australian people, well, look, we're seeing a bit more of these mild reactions, but our analysis says that it's still quite safe to take these vaccines.
So Alan, just another question. Will people get a choice? A lot of people are asking that question. Will they be able to choose their vaccine? Well,
in the initial phase, no, because there's only Pfizer vaccine in phase 1A until AstraZeneca arrives. And then after that, there will be Pfizer hubs and there will be AstraZeneca hubs, but in general, no, it'll be rolled out at those centres, and I guess you can try and pick where to go and get your vaccine, but there'll be much more AstraZeneca vaccine available than Pfizer vaccine available. I don't know, Paul, if you want to add to that.
No, I just typed an answer to that one too. It's pretty much the same, Alan. There's always a choice insofar to a certain extent, but the reality is for Australia in 2021, most people are going to get the AstraZeneca vaccine if they want to get the vaccine. It's not compulsory, but if you want the vaccine, AstraZeneca is the most likely one that you will be offered. Pfizer is starting, but only in relatively small amounts coming in over the course of 2021. Novavax not yet registered by the TGA, nor is it available in Australia. So waiting for that one will mean well after winter, I would say. My advice to everybody is to, whenever your chance comes up to be vaccinated, to get two doses of the vaccine as quickly as possible. And particularly if you're in a vulnerable group, or for patients of yours, or of those people online that are in those groups.
Thanks, Paul. I'm not sure who would be best to answer this question, so I'll just throw it to the floor, but it's the second most popular question. Why is it that there's an accreditation process for GPs to be able to deliver this vaccine? And why are pharmacists being included in the vaccination program?
That's probably the government guy, so I'll talk to that. So we have gone for accredited GPs because they're accredited, and I think there's been a very long period of working towards accreditation for GPs, and that relates to the Medicare benefit schedule. There is a very generous payment that will be made to GPs, which will be boosted if accredited general practices also give the second dose to an individual. So that's the way we've worked through the payment schedules for GPs. Of course, GPs are not the only people that will be providing vaccinations. The states and territories will have their Pfizer hubs starting on Monday, and there will be AstraZeneca also provided to the states and territories, and they will have their own mechanisms for filling in the gaps, if you like, in areas where they need to do that, whether that's people that don't have GPs or places where GPs may not be present. Pharmacists have for many years been involved with immunisation in Australia, particularly every flu season, but in some states and territories involved with the National Immunisation Program. So that's a way of increasing access. Many people choose to get their flu vaccines through pharmacists. And so they won't be involved immediately. They won't start until into phase 2, which will allow us to really get good understanding of the vaccine in the Australian context, but they will be included, so we can increase the access to people. We want it to be free and available to as many people who want to get vaccinated as possible throughout 2021.
So another question for you, Paul, that's also quite a popular one is, what are the plans to tackle vaccine hesitancy, and do we have any plans for promotion like they're doing in the UK with Elton John and Michael Caine?
I love that that advert. Any suggestions? You can put it in the Q&A as to who would be the most influential individuals, but we have some ideas. Yes, there is a large communication campaign which has been launched, but not sort of blanket campaign at the moment. Obviously, we don't want to build too much expectation when, in fact, it's impossible for at least another couple of days to get a vaccine in Australia. But as that rolls out, and particularly when AstraZeneca comes on board, particularly when we increase the access through GPs and others, you'll see a lot about the vaccine. We've had specific engagement, really strong engagement with our culturally and linguistically diverse community and the Aboriginal and Torres Strait Islander community, working through specific campaign messages and looking for the involvement of elders, of community leaders, religious leaders, and so forth. There's been a fatwa that has been pronounced by the Imams of Australia to say that people should get vaccinated. It's the right thing to do for your own health, your family's health, for the wider community, and so that was a wonderful thing. So we're involved with churches and other community groups. So yes, there's a lot of that. Of course, we're having a lot of these sort of webinars and trying to amplify the messages through all of you. And again, I'd come back to all of us to work as a community that's interested in immunisation to work with those hesitant groups and to get the real story out there to counter some of the fake news.
Thanks so much. I would love to keep going with questions, but I think we've reached time, so I'll hand over to Christine.
We have reached time, and I'd really thank our audience for staying with us on this Friday evening, and how compelling our speakers have been tonight. I would actually ask our audience if they want to leave any comments or questions about future webinars that you'd like to see. Certainly, I think we would like to touch on the very important topic of vaccine acceptance, uptake and demand. And I would absolutely echo the comments that Paul made about the importance of mobilising community champions and you know, how important it is to engage with all groups in Australia to ensure that we have the most equitable program possible, as well as, of course, the contribution to the region. But Paul, I'd like to thank you immensely for being on the spot with us tonight, for answering so many questions, and also very much thank Alan Cheng, Hao Cheng and Michael Nissen and Katie Flanagan, as well as Ket for their roles. And of course, Andy Pollard, who's had to leave us a little bit early this evening, but was absolutely wonderful to hear from. And we look forward to joining you for the next webinar series in our series very, very soon. In the meantime, good evening, and let's look forward to Monday when the first vaccine doses in Australia will go into arms. Thanks very much, everyone.
Thanks, Christine.
Thank you.
Good night, everyone. Thank you, Christine.
Thank you. Good evening.